Okay, here we go. New Cyanotoxin Guideline values and guidance for controlling occurrence. These are the 5 issues that I want to mention in my talk, but I want to start out by thanking you very much for inviting me to do this. It's going to be really exciting for me to see what's going on in US EPA and in the United States, and I'm really looking forward to the rest of this meeting. I want to start out with a very short introduction, assuming most of you know quite a lot about cyanotoxins, so I don't really need to tell you a lot about them. I want to spend most of the time for this talk on the new guideline values and how they were derived, and then differentiating by short-term interventions. How can we monitor? How can we react quickly if we see that blooms are going in a direction where we need to do something to prevent people from getting exposed? And then point 4, water safety planning. What can we do for longer term, appropriate and effective risk assessment and monitoring? And I'd like to finish with a very short bit on communicating risk to recreational site users. Some of you are familiar with this book. This is the new version, and I think I'll hold it into the screen so you can see the full 850 pages. I want to emphasize that you can download it for free from the WHO website, and you can buy it, of course, if you want to have it in your bookshelf. I'd like to start out with calling our attention to the toxicity of cyanobacteria. Per unit biomass, they are about as toxic as very toxic mushrooms. Of course, mushrooms are a lot drier. Cyanobacterial scum is quite a liquid affair, so it's quite hard to swallow as much as would have acute effects immediately, like with mushrooms. You're all aware of this list of cyanobacterial toxins, I'm sure. I like to point out that in toxic cyanobacteria in water, we have a short subsection on lipopolysaccharides where we actually ventured to suggest that they are not really an issue with cyanobacteria for 2 reasons. One is that the research that's been done about them for a couple of decades now has shown that they're not nearly as toxic as the lipopolysaccharides from Salmonella. But the perhaps more important argument is Salmonella multiply in people, but cyanobacteria don't. It would be very hard to get as high a dose into your system that would cause the huge inflammatory response that we get from lipopolysaccharides produced by Salmonella as they multiply in our guts. So we think they are not really an issue and maybe should be removed from the list of what we call the cyanotoxins. This is an old overview of occurrence, but I think it's still what we see typically in surveys. If not, if you have different experience, I'd be really interested in knowing that. One key message here is that microcystins occur in more than half of the blooms and water bodies that we monitor, even if we have a fairly even distribution of water bodies monitored across trophic state. And we've seen something similar with cylindrospermopsins in Germany, although they are rarely in high concentrations, quite in contrast to microcystins. And the neurotoxins, we don't have any data on anatoxin A/S. It hasn't been monitored much, but anatoxin A and the saxitoxins don't appear to occur nearly as often. A short overview of the concentrations, and you can easily see that the very high concentrations that we get in scums of microcystins and sometimes with anabina, but not quite as high, is not quite what we see with the neurotoxins. And especially with cylindrospermopsin, very high concentrations are also quite rare. But the important thing with these is that up to 90% of the concentration can actually be dissolved in water. And that is not what we see with the other cyanobacterial toxins. So that means we have a different type of challenge with drinking water treatment. It's not enough to just filter out the cells, in some cases at least. And it seems they can be quite persistent, and I'll be showing you a graph about that a bit later. A few words on BMAA, because I was notified to that being an issue for the work of some of you. BMAA has been discussed hugely emotionally, and things have happened such as a publication reporting data from fluorescence detection and actually stating that this was confirmed with MS, but then you look at the supplementary material where the MS data are shown and you find only a very small concentration. And the point here is that wherever, as far as I'm aware, BMMA has been analyzed with a more recent, very selective analytical methods, concentrations have been extremely low. And the animal experiments that show effects have been done at hugely unrealistically high exposures. So with appropriate methods, there doesn't really seem to be an effect. And neurologists state that the four quite distinctively different neurological disorders that have been associated with BMAA are not really likely to all be caused by the same substance. So that's the key message in the chapter in the new book about this substance. So I need to get pictures out of the way, sorry. So on occurrence, the point I'd like to make is that we haven't identified any new groups of cyanotoxins since cylindrospermopsin in 1992. It seems we might actually know the most important groups, although we still occasionally see reports of effects that cannot be attributed to the concentrations of the known toxins in the sample. Microcystins are very likely with surface scums of microcystis. Quite rare to find a scum that does not have microcystins. And the same applies to Planctotrix agardii, the Planctotrix that's homogeneously distributed in the water. It can reach really quite high concentrations as well, in the range of a couple of hundred micrograms per liter. And the toxins cylindrospermopsin, anatoxin A, or saxitoxins produced by Nostocalin, nitrogen-fixing cyanobacteria, seem to occur less frequently and at less high concentrations. Now, to move on to the new guideline values. First question, if you have human data that you can use for guideline derivation, that's always a good way to go. A point that we really want to make, however, is that the mere co-occurrence of cyanobacteria and then very unspecific symptoms like skin irritation or gastrointestinal irritation is scarcely indicative of the known cyanotoxins actually having caused these symptoms, and more likely that other yet unknown agents in the water, and that doesn't even have to be cyanobacteria, it could be a lot of other things, would actually be the more likely cause. Now, if the symptoms are toxin specific and you also have high concentrations of that toxin, that's a bit harder evidence. And if you find cyanotoxins or cyanobacteria in the stools of patients or toxins in body fluids, that's a very good indication of exposure, of course. But again, a problem here is that we don't really have a ratio to effects. We can do that with oral exposure, but we cannot do the body tissue concentrations. We don't know what concentration would relate to which effects. So even that kind of data aren't really useful for deriving guideline values. The epidemiological evidence is also pretty messy. For recreational exposure, we have contradictory studies. Some find some slight effects and usually not toxin specific. And even one and the same author found a relationship that was statistically significant in the first study that Toledo did, but not anymore in the second bigger study. Exposure through drinking water or food, a lot of research, especially on cancer promotion from China, is available. But again, this is indicative because we have huge problems estimating the concentrations to which people were exposed. And if people were exposed to surface water, of course, there are a lot of other contaminants in the surface water as well, possibly including, for example, pesticides, which also include some carcinogens. And the demographic information, how long have people lived in the place, where have they been exposed, to how much have they been exposed, all of that is nothing that you can use for quantitative guideline derivation. In fact, where WHO does use epidemiological evidence for guideline derivation, it's usually from occupational exposure, where you have people cohorts of people where you actually can trace what they have been exposed to a lot more closely. A further increasingly popular approach is in vitro laboratory studies with cell lines, transporters, enzymes. And the nice thing about these is that they help you understand the mechanisms of toxicity. The problem, again, is that you cannot quantify. It's at best a qualitative approach. And we might get to the point where you can roughly put chemicals into risk categories, but because you're just looking at one small segment of the whole pathway of cause-effect, you cannot very well quantify. So we are back to the data from toxicological testing in the lab with whole animals. Not such a nice thing to do, not very popular, and increasingly reduced in laboratories. Hard to get the data. We get two different types of responses. We get substances with a threshold for toxicity, and that's actually most of the substances that we regulate in drinking water, or substances without a threshold where you can just look at a risk of getting usually cancer. Many of you who work with toxicology probably understand quite well how we derive tolerable daily intake levels. I still like to show this just to show people very clearly how the dose that we tolerate ends up relating to a dose that actually causes demonstrable illness or harm in the experimental animals. So to move from leaving out this big red box and magnifying the lower ones a little bit, the no observed adverse effect level in the experiment done with mice in the 1980s and published in the 1940s and 1990s by John Fowle actually came up with a dose that had no observed adverse effect of 40 micrograms per kilogram body weight and day of these mice. And that then gets divided by a factor of 1,000 for a factor of 10 for the differences between species in their sensitivity, another factor of 10 for differences between individuals, and another one for uncertainty of the database. And that gets you to this tolerable daily intake, which is really a tiny fraction of the no observed adverse effect level. And that is then, that is then multiplied by the mean body weight of an adult and a proportionate factor for the share of exposure via drinking water, where WHO uses 0.8, and 2 liters of water per person and day to get you to the 1 microgram per liter for drinking water that you are all aware of for microcystins. Now, I've shown you the Toledo photo 2 times already by accident. This incident where the exposure through drinking water was actually at a level around 2 micrograms per liter and only for a very few days stirred up a lot of attention. It hit the media in Europe as well, and awareness in the World Health Organization that short-term guideline values would be very useful to avoid panic in situations like this, where measures like don't use for cooking and showering, closure of restaurants and shops, schools closed. In COVID times, I don't need to tell anybody what that means for society, and that's probably not a good idea to overdo the response to that kind of a situation. Although in the longer term, of course, it was good that Toledo upgraded their drinking water treatment system. So WHO came up with short-term guideline values, and the difference to the lifetime guideline values is that the uncertainty factor is removed when deriving the tolerable daily intake. And that is very justified because the experiments with mice were done over quite a number of weeks, I think 14. And here we're talking about a short-term value that could be tolerated for at most 2 weeks, and not every other 2 weeks in the season, but once a season. The idea is to give people time to respond in a more effective manner. And the other difference is that the allocation or partitioning factor is 1, because in such a situation, exposure for the whole population would mostly be through drinking water. And that then got you to 12 micrograms per liter as the short-term drinking water value for daily exposure for at most 2 weeks. For recreational exposure, there's a slight difference in the assumptions in that they used the same tolerable intake, but they used a 15-kilo toddler consuming a quarter of a liter of water, and that got to the value of 24 micrograms per liter. The other good news is that we now also have a guideline value for cylindrospermopsin in drinking water, and we have a short-term value for both cylindrospermopsin and for microcystins. And for saxitoxins, the data that were available from food exposure took us to a acute value, and that does not mean every day for 2 weeks. It just means for a single exposure of 3 micrograms per liter, and that is for babies. And for anatoxin A, the toxicological database was just sufficient to give an upper bounding value that they called health-based reference value of 30 micrograms per liter. And these are the corresponding values for recreational exposure. And you can look them up on this website or also on the one in the link that Shane just said was sent to everybody who's participating here. Now, a few take-home sentences that if we talk about hazardous substances in surface waters, I think cyanotoxins are probably the most widely occurring by far, especially as long as so many of our water bodies are eutrophic. And we can consider them as natural substances, but the fact that they occur in these high concentrations is in very many cases not natural. All in all, however, acutely toxic concentrations actually of any substance are not very likely in drinking water. For cyanotoxins, I think they're more likely in recreational water where we use water bodies with heavy scums. But we can also remember that short-term exposure to a few micrograms per liter is most likely to be quite harmless. And that cutaneous exposure to cyanobacterial blooms, although it's probably the cause of some of the mild self-limiting symptoms that have been reported, we are not sure whether the cyanobacteria are really the causative agents. And as I said, they're mild and self-limiting. On the other hand, cyanotoxins may cause high risks and even death through ingestion or aspiration of high concentrations. We have very few cases of known human injury or fatalities. The ones that we do know that are clear are through hemodialysis. But on the other hand, that's not to be expected, especially not in drinking water where we regulate chemicals so that risks are extremely low. And also not in recreational water bodies because chemicals are typically not regulated because of chronic and acute, not because of acute risks, but they're regulated because of chronic risks in drinking water. And I think in recreational water bodies, the health risks, the acute health risks are primarily from pathogens. Now, some words on using what we know about guideline values for short-term interventions. Of course, whether or not we have toxins in the water depends on whether we have cyanobacteria, which depends on nutrient concentrations in the water body, mostly phosphorus concentrations, which depends on catchment loads. And we have this whole cascade of cause and effect that we can address. And if we're not successful in addressing that, then we need these short-term responses for drinking water and for recreation. We started out 20 years ago with the first edition of toxic cyanobacteria in water at a point where monitoring toxins was expensive and not accessible for everyone, and that still is true for many parts of the world. So some indicators, surrogate parameters are important. And one option is to use biovolume or another surrogate such as chlorophyll A concentrations. And we suggested in the book to use them on a level that would reflect a worst case. Now, if you look at microcystin per unit biovolume in cultures, you can get concentrations up to 5, 6, 7 micrograms per cubic millimeter of biomass, biovolume. In the field, in natural samples, they rarely exceed 3, this ratio of biovolume to microgram, to cubic millimeters of, sorry, again, the ratio of toxin to biovolume. So we suggest to use a ratio of 3:1 for microcystin to biovolume, or 1:1 for microcystin to the concentration of chlorophyll A as a worst case approach. In real situations, it's often 1/10 of that. So it's, if you have the options to analyze toxins, it's certainly worthwhile to do that, because you may be able to tolerate substantially higher biomass before you need to intervene. And of course, WHO just gives recommendations. It's nothing prescriptive at all. And the book very much encourages people to develop own toxin to whatever parameter is useful ratios, toxin to turbidity, toxin to fluorescence, toxin to a remote sensing signal, whatever works best locally. Now, we think that for other cyanotoxins, the ratio that we derive from microcystins is sufficiently protective also, for one reason, because we know that the concentrations are rarely as high, and because that, well, we know from culture work also that we have roughly similar ratios in cultures. And also, I need to emphasize that the only one where this will not work is cylindrospermopsins, and that's because of the quite high share of extracellular toxin. And this graph shows you the development over time of the probable producer organisms in this case, and the cell-bound toxin and the toxin dissolved in water. And you can see that it was hanging around in this lake even late into autumn, while the producers were already gone. So for cylindrospermopsin, monitoring the concentration of the toxin directly might be more important. We derived 3 different levels of alerts to which one should respond. And I think I'd probably go into those directly. And the alert level frameworks that for drinking water you're already familiar with, perhaps, if you use the suggestion in the first edition of Toxic Cyanobacteria in Water. It suggests to start out with looking at your water body, visual inspection, and seeing whether you see cyanobacteria or strong turbidity. Check in the microscope who done it, what's the cause, is it cyanobacteria? And if you see a significant share of cyanobacterial cells, and that's before you even start counting, that takes you to the vigilance level, where you could intensify monitoring for cyanobacteria, inspection of the lakes or drinking water offtakes. And then you can either look at biovolume or chlorophyll A as indicator, or if you have the option to look at toxins, that's fine. And if you're below the lifetime guideline values, that takes you back to the vigilance level. If you're above that, it takes you to the alert level, and that asks you to check whether you can do something about getting cyanobacteria into your drinking water offtake. And if not, it takes you on to informing the public, to consulting with health authorities, intensifying monitoring. And if you're then above the indicators for exceeding the short-term guideline values or above the short-term guideline values themselves, you're at alert level 2, where you really need to do something, you need to intervene. I'm going to skip this because of time. The same system is suggested for, or a similar system is suggested for recreational water, but with a somewhat different subdivision. One branch that you can use is only site inspection, visual site inspection, and that's not only for poor countries who don't have the means to do more than that. It might also be very important for health authorities, responsible authorities who have many, many, many bathing sites to look at and just simply can't do a lot of monitoring and a lot of measuring. Or as other option, or both options supporting each other by lab analyses. And again, starting out with microscopy, with looking at who's there, who can be producing the toxins, and looking at levels of toxins or levels of biomass. And if you're above them, that then again takes you to different alert levels and to using the recreational guideline values for decisions. Watching out for time a little bit, because I do want to have a few minutes for water safety planning and the longer term, not end of pipe, not last minute measures, but what you can do to prevent blooms. That takes us to this part of the story. And that's where the World Health Organization promotes developing a site-specific water safety plan for your specific situation. This is the overview that I want to show a bit close up. The first step, getting ready for the assessment and for planning, asks for forming a team to do the job. And I think that has a lot of charm specifically for toxic cyanobacteria, because to control them, you need a lot of different expertise. You need limnologists who understand the water body, and you need catchment managers who understand the pathways of nutrients to the water body, but you also need the stakeholders for activities in the catchment if you want to get them on board with controlling these activities and controlling the nutrient loads. And I think the water safety plan team provides an opportunity to create a platform where these people can talk to each other and where you can perhaps have a chance of motivating some of the stakeholders in the catchment for the measures that you develop together and where they recognize how important this can be. A core of developing a water safety plan then is describing the water use system. And if you talk to people, you'd be surprised how often a water utility does not have a good description of their system. It's certainly not documented, often in people's minds, but not in the documents. And then assessing how high the risk is and developing measures to improve the situation. Monitoring in the context of a water safety plan focuses on monitoring whether the control measures in place are effective. And that can be as simple as a fence around a reservoir to keep the cattle out. And monitoring would be checking whether the fence is still intact. And of course, documentation, developing supporting programs like training staff and periodic revision and review of the whole plan. A short look at assessing the risk. Let's take the example of a slowly flowing large river supplying a population of a town of 500,000 with occasional massive microcystin blooms, sometimes lasting over 3 months during drought, and no other option for raw water offtake, and quite normal drinking water treatment, but not in best condition, with occasional breakthrough, no ozone, no activated carbon. You'd come up with a risk assessment. Okay, we can take out the cells quite effectively most of the time, but we may have some breakthrough. It's probably not a major health impact because we wouldn't be above the 12 micrograms microcystin short-term value, but we'd probably be above the lifetime guideline value occasionally. Exposure through recreational contact, probably not much of a problem because swimming is banned in this river, too much shipping traffic. Contaminated foods, probably not a problem. Okay, few people go fishing, but we've been discouraging that. Hardly any irrigation directly with this water, so that's not a big issue. Now, of course, in real life, you wouldn't be looking only at cyanotoxins. You'd be developing a water safety plan for the other hazards as well, and you'd come up with other things like microbial contamination of the river in this messy situation, or even Legionella in people's household installations, or for recreation, drowning injuries because of the illegal swimming in the boating channels. And of course, you can always discuss the objectivity of assessments like this, but the experience shows that people who do this come out of that exercise saying, okay, we're not totally sure whether we placed everything in the right box, but we know why we did it. We have a much better understanding of our system.
Hi, Ingrid. We have run out of time, and we have a couple of questions.
I need 1 more minute to finish. Is that okay?
Sure. Yeah, that's fine.
This slide was just to show you that these kind of assessments would be very different for these three very different systems. I won't go into it, but I want to emphasize the message that a water safety plan is always something quite site-specific, and it has the charm of including the uncertainty of the assessment. In this example, people said, we can't make a decision yet. We need to quantify the cyanobacteria first. I told you about the benefits. I was asked to say something about communicating the risk to users, and I think we were quite successful with this very simple message, showing people what to look for. Let's skip that one. I want to ask you to stay safe. I want to tell you that this is the structure of toxic cyanobacteria in water, with chapters giving you a lot of scientific background and with what I just went through about exposure through drinking water and recreation, but also with this whole pyramid of control levels and a section on the practical aspects of monitoring, analyzing cyanobacteria and the toxins. And I'd like to thank you for your attention.